For those who are interested. The title of her talk is Not Just an Empty Shell, 6 Centuries of Shellfish Exploitation at the Hane Dune Site, Marquesas Islands.
Okay. Bonjour à tous, c'est Alana. Donc, je m'appelle Gabrielle Traversa et je suis doctorante à l'Université de Paris 1. Et aujourd'hui, je vais vous présenter le cas d'étude principal dans le cadre de ma thèse qui porte sur l'exploitation des mollusques et des échinodermes sur l'île de Huka, aux Marquesas. Donc, comme ma collègue Vahine et RURUA, ma présentation va porter sur le site de Hane, mais ici, donc, je traiterai des mollusques. Donc, ma collègue vous a précédemment présenté le contexte géographique et archéologique du site, donc je ne vais pas revenir dessus. Tout ce que je dirais, c'est que mon assemblage provient aussi de la fouille de 2009 qui a été menée par Éric Conte et Guillaume Moll dans le secteur nord donc, de la fouille des années 60, sur une surface de 18 mètres carrés et avec une stratigraphie d'une profondeur de 2,60 mètres. La nouvelle fouille a, encore une fois, permis l'établissement d'une des séquences chronologiques les plus longues pour la Polynésie française, attestant d'une occupation humaine entre le XIe siècle de notre ère et le XVIIe siècle. Donc, la faune du site a précédemment fait l'objet d'une étude de la part de Patrick Kirsch dans les années 70, à partir de l'assemblage donc, de la fouille de Sinoto et Kelon. Les quantifications étaient réalisées par poids des restes et les restes étaient identifiés par grande famille. Dans son étude, Patrick Kirsch identifie une augmentation constante des quantités de mollusques au cours de la séquence et il identifie quelque chose qui est assez intéressant, c'est la diminution abrupte des quantités de chitons ou polyplacophores, qui est une classe de mollusques, à partir de la première phase d'occupation du site. Il identifie, enfin, en tout cas, il lie cette observation avec la diminution des restes d'oiseaux et de tortues au cours des premières phases d'occupation, ce qu'il interprète comme une surexploitation des ressources vulnérables à partir de l'arrivée des premiers Polynésiens. Donc, pour étudier mon assemblage, j'ai effectué un travail de terrain sur l'île de Huka afin, d'une part, d'observer les biotopes et le littoral marquesien qui se caractérisent à l'inverse du reste de la Polynésie française, par une absence totale de lagons et de récifs barrières, ce qui induit donc une biodiversité très particulière. J'ai également effectué des entretiens avec les habitants de Huka afin de discuter de la place des coquillages dans leur quotidien et de discuter des pratiques actuelles de collecte. J'ai également moi-même réalisé des collectes de spécimens frais pour mettre en place une collection de références qui pourra m'aider pour mes identifications. De retour au laboratoire, après une revue des données ethnographiques, ethnohistoriques et archéologiques sur les marquises à propos des coquillages, j'ai donc axé mon analyse sur une méthodologie archéomalacologique, qui est une discipline qui a connu de nombreux développements ces dernières décennies. Donc j'ai identifié taxonomiquement au rang le plus précis possible mes restes. J'ai utiliser plusieurs méthodes de décompte comme le nombre de restes, le nombre minimum d'individus et le poids des restes par gramme. J'ai également observé les altérations taphonomiques des restes, qu'elles soient naturelles ou anthropiques. J'ai réalisé des mesures biométriques et enfin, j'ai fait de l'analyse spatiale des restes sur le site. Donc l'assemblage de Hane est composé d'un total de plus de 26 000 restes pour 1702 individus identifiés et un poids total de 13 kg. Le spectre malacologique est composé de 57 gastéropodes, 14 bivalves, 2 polyplacophores et 3 familles d'oursins. C'est donc un assemblage qui est composé de nombreuses espèces où on a quelques taxa qui dominent. Notamment, la première, la petite patelle ou limpet, Selana radiata, et le polyplacophore, Selana radiata, et le polyplacophore Acanthoplora gemata. Ces deux espèces sont suivies de très loin par l'autre polyplacophore Chiton marquesanus et d'autres gastéropodes comme le turbo ou les purpuras, plusieurs espèces de porcelaine ainsi que quelques bivalves comme les huîtres et quelques mous. Donc j'ai pu, à partir de ces identifications, noter une répartition des zones littorales et des substrats. Donc j'ai noté une prédominance d'espèces vivant en zone intertidale et à l'interface intertidale-subtidale, ainsi qu'une exploitation ciblée des substrats rocheux, ce qui est en accord avec le littoral marquisien qui propose de nombreux platiers rocheux ou ce qu'ils appellent trottoirs marquisiens et comparativement très peu de fonds sableux. J'ai également une répartition des gastéropodes qui est beaucoup plus élevée que des bivalves et là encore, c'est en accord avec la diversité malacologique de l'archipel qui propose 7 gastéropodes pour un bivalve. Pour ce qui est des quantités, des données quantitatives au cours du temps, j'ai pu noter une exploitation des mollusques beaucoup plus intensive lors de la première phase, qui est donc la première occupation du site, et notamment au niveau H, où spatialement les restes sont répartis sur toute la zone de fouille, ce qui est moins le cas, ou en tout cas moins visible, lors des 2 niveaux précédents. À ce moment-là, les polyplacophores constituent la majorité des individus identifiés. À partir de la deuxième phase, les quantités diminuent de manière constante, donc à l'implantation du hameau côtier, et à ce moment-là, les gastéropodes sont les plus nombreux, dominants en termes de nombre d'individus. On peut voir donc ici l'abandon du site pendant les niveaux D et C, et ensuite le niveau des inhumations, donc de la reconversion du site en cimetière, où on a des quantités qui sont relativement similaires à celles de la phase 2, que j'interprète comme une fréquentation sporadique de la dune à cette époque. Mais spatialement, les restes ne sont pas liés aux inhumations. Le résultat principal de mon analyse, c'est l'exploitation ciblée au cours de la première phase d'une espèce particulière de polyplacophore qui est Acanthoplora gemata. C'est une espèce qui peut atteindre jusqu'à 12 cm de long, donc c'est une espèce intéressante en termes de chair disponible. On voit qu'elle représente jusqu'à 57% du NMI au niveau I et un maximum de 151 individus au niveau H. À partir de l'établissement du hameau côtier, les quantités de cette espèce diminuent de manière drastique pour ne représenter plus que 10% du NMI total au niveau E, donc en fin de phase. À partir de ce moment-là, c'est la petite patelle, Selana radiata, qui devient l'espèce dominante de mon spectre malacologique. On voit d'ailleurs que les anciens marquisiens n'ont pas remplacé une espèce de chiton par une autre, donc l'autre c'est Chiton marquesanus et qu'on voit en gris ici, mais se sont tournés vers une ressource qui est beaucoup plus petite, puisque mes patelles ne font pas de plus de 3 cm de long sur toute la séquence, mais extrêmement nombreuses sur les nombreux platiers rocheux qui abordent le site. Donc j'ai réalisé des mesures biométriques sur toutes les plaques entières, antérieures, intermédiaires et postérieures de chiton. Et j'ai pu noter, pour ce qui est des plaques postérieures, où j'ai gardé la longueur totale de la plaque, une certaine diminution des moyennes au cours du temps. Il en est de même pour les plaques intermédiaires, où j'ai gardé là la largeur de la plaque. Et on note également une certaine diminution des tailles entre le milieu de la première phase et le début de la deuxième phase. Ce qui est intéressant, c'est que sur les autres sites que j'ai pu étudier dans le cadre de ma thèse, j'en ai étudié 4 autres, aucun ne contient de Acanthoplora gemata. Tous ont par contre des quantités de Chiton marquesanus qui sont relativement importantes. Ce que j'ai noté aussi, c'est que les habitants de Huka aujourd'hui consomment encore beaucoup Chiton marquesanus, mais la large majorité d'entre eux n'ont jamais vu Acanthoplora gemata. Et ceux qui l'ont vu, notamment les personnes les plus âgées, ne le considèrent pas comme une source de nourriture. C'est une observation qui est partagée par les biologistes marins avec qui je me suis entretenue et qui considèrent que cette espèce est limitée à l'îlot de Heiao, qui est une toute petite île inhabitée au nord de l'archipel des Marquises, mais aucun n'en ont vu sur les autres îles de l'archipel. C'est relativement intéressant. Donc pour résumer la question de la subsistance, les résultats par rapport aux polyplacophores répondent assez bien aux observations qui avaient été réalisées par Kirsch dans les années 70. C'est d'ailleurs un cas de figure qui a été également observé par Barry Rollett sur l'île de Tauata et le site de Hanamiai. Mais cette fois, les quantités de chitons sont de l'espèce de Chiton marquesanus et diminuent de manière drastique à partir de la première phase d'occupation du site. Par contre, pour ce qui est des données quantitatives générales, mes résultats ne montrent pas d'augmentation de quantité des restes sur la séquence, et justement, ils montrent l'inverse, ce qui est dû à la nouvelle stratigraphie du site de la fouille de 2009, où on voit très clairement un abandon du site, puis une reconversion et un changement de fonction du site. Donc, outre la question de la subsistance, je vais vous parler plus rapidement de la technologie, puisque les communautés de Hane ont exploité les mollusques pour leurs coquilles afin de façonner divers types d'artefacts, notamment les engins de pêche, donc les hameçons et les leurres à bonites, différents types d'outils et des éléments de parure et des instruments de musique. Donc l'espèce dominante en termes de technologie, c'est Pinctada margaritifera, qui est l'huître perlière. On voit que son exploitation a été la plus intensive à la phase 1 et au tout début de la phase 2, et notamment au niveau H. Son utilisation est axée majoritairement sur le façonnage des engins de pêche, mais je ne me suis pas intéressée à ça, puisqu'un travail a déjà été amorcé indépendamment de ma thèse. Par contre, ce que j'ai pu trouver, c'est des charnières travaillées dont la partie supérieure et la partie extérieure a été polie et que j'interprète comme de potentiels couteaux ou peut-être des scrapers qui sont également identifiés dans la littérature ethnographique et archéologique. Parlant de travail de la nacre, j'ai également retrouvé des limes en radiole d'oursin, l'oursin crayon, qui sont présents uniquement en phase 1 et c'est intéressant puisqu'ils semblent complètement remplacés par les limes faites en branches de corail et c'est quelque chose qui a été également noté par Sinoto et Rolette sur leurs sites respectifs. Donc, nombre de ces espèces se partagent un rôle alimentaire et technologique et ont pu être récoltées au même moment. C'est le cas de Mauritia mauritiana, qui a servi à faire les objets très connus des marquises, qui sont les pelle-fruits ou iime. J'ai pu en retrouver et j'ai voulu retracer les différents gestes qui rentrent en jeu dans les étapes de façonnage de cet outil-là, à partir de mes propres observations et de celles de Marie-Marie Kellum dans les années 70. J'ai également retrouvé des tests de Purpura persica, uniquement en phase 1, donc percés sur la partie dorsale, et plusieurs auteurs en archéologie suggèrent que ça aurait pu être des types antérieurs de pelle-fruits qui auraient été remplacés progressivement par l'utilisation des porcelaines. Enfin, je finirai sur des éléments de parure qui sont uniquement présents en phase 1 et en tout début de phase 2, qui sont faits tous dans la même espèce, Naria elvola. C'est quelque chose également que Barry Rolette a relevé sur son site à Tauata. Et en étudiant les altérations taphonomiques, j'ai pu en déduire que ces coquillages-là ont tous été récoltés sur la plage à l'état d'épave. J'ai d'autres coquilles travaillées qui sont un petit peu plus difficiles à interpréter, comme cette valve d'Atrina pectinata sectionnée. C'est d'ailleurs la seule occurrence de cette espèce sur le site et sur tous les autres sites de mon étude. Donc, le site de Hane est un site qui est très riche et qui présente un cas d'étude très intéressant au niveau de l'exploitation tant alimentaire que technologique des invertébrés marins par les anciens Enata. Il m'a montré une collecte large de mollusques intertidaux disponibles sur les nombreux platiers rocheux de l'île. J'ai également voulu montrer que grâce à des outils méthodologiques actualisés et plus précis, il était possible d'avoir une meilleure résolution des interactions de communautés humaines anciennes et leur environnement, et avec les invertébrés marins tout particulièrement. Par la suite, j'aimerais pouvoir développer plusieurs points de mon travail. D'une part, compléter ma collection de références afin de potentiellement tester des reconstitutions de taille, notamment sur les chitons. J'aimerais également réaliser des analyses plus précises au niveau microscopique, des analyses de microtraces, d'usure et de résidus, ainsi que plus largement étudier d'autres assemblages archéomalacologiques dans les différents groupes de l'archipel des Marquises et plus globalement en Polynésie orientale afin de mieux appréhender l'exploitation des invertébrés marins dans différents contextes, d'îles hautes avec lagon et d'atolls avec lagon ouverts ou fermés. Maruru, Koutonui, merci.
Our next talk is going to be by Patricia Pillay, A Day in the Life of Kuri, the Maori Dog in Aotearoa, Insights from Zooarchaeology.
Yorana, bonjour, kia ora koutou, a warm greetings to you all. I'm going to talk about my recent master's research on the diet and dental health of the Māori dog, or kuri, in Te Ika a Maui, North Island in Aotearoa, New Zealand. So, the Polynesian dog that accompanied the first ancient Pacific navigators to Aotearoa from the mid-13th century are known to Māori as kuri and have an undeniable significance in Māori culture and history. In Māori oral traditions, Pacific explorer Kupe, often credited with discovering Aotearoa, is said to have carried his dog with him on the long and challenging voyage from Hawaii. Other historical accounts and traditions highlight the significant role that dogs held in Māori society prior to European arrival. And across the world, there is a range of evidence that highlights how dogs, the oldest animal domesticate, have captivated human society for millennia. To give but a few examples, dogs are seen as companion animals, hunting aids, for haulage, as sources of wool and food, as well as ritual activity. Outside of Aotearoa, a strong focus on domestic dogs is encountered in indigenous contexts. This includes specific detail on their appearances, as well as such as the well-known Seshat dogs or wool dogs in the Pacific Northwest Seshat traditional territories. Similar to kuri, the hairs from these dogs would produce wool and textiles, such as blankets worn during rituals and ceremonies, and often traded and gifted as prestige items, as well as for regular use. So returning back to Aotearoa, we see similar yet unique aspects of dog husbandry and interactions with Māori. The archaeological record can further highlight the importance of dogs in Māori society as a source of food, their fur used for dog skin cloaks, bones as artifacts, and they are depicted in rock art and carvings as shown. And the movement from the small islands of tropical Polynesia through to the vast and varied Aotearoa New Zealand landscape presented altogether novel opportunities for human-dog relationships to move in new directions. And dogs were widely dispersed throughout the country during the colonization process of early ancestors of Māori. And although dogs are highly valued, dog bones and teeth are often uncommon in Pacific Island archaeological sites. And that suggests that they were likely kept in small numbers. And on some of the region's smallest islands, they were initially introduced but then disappeared rapidly over time. And we also know that Polynesian dogs in Hawaii had caries or cavities from a diet rich in starchy carbohydrates. So it's interesting to see how kuri compare in relation to these other Polynesian dogs. So that brings me to the remarkable distribution of dogs in Aotearoa. You can see from my survey of dogs through the cultural sequence that they rapidly dispersed across the length and breadth of the country within the first 200 years of human arrival. They're also found in early human occupations on many large offshore islands, including the Auckland Islands. The distribution maps showcase some distinctive patterning where kuri are most abundant on Te Waiponamu South Island, specifically along the eastern seaboard, where settlements map onto areas rich in large fauna, or which might provide access into the interior areas of prime moa habitat. However, kuri are also well represented in the subtropical North Island, though in reduced numbers relative to Te Waiponamu. They were, however, particularly abundant at the 14th century settlement of Hauhora in the Northland, where moa hunting was an important activity, but also the use of dogs. And you will see that there are a reduced number of dogs between about 1450 AD and 1650 AD, which is another interesting aspect, but we won't get into in this talk. However, you can see a marked increase in dogs after 1650, with kuri again well represented on both the North and South Islands. And this late pattern is consistent with observations of the earliest European visitors who describe instances of kuri accompanying Māori in varied contexts. And kuri are still seen to be relatively abundant from the archaeological remains through till the 19th century, and these distributions align quite well with Māori histories. Some of these sites include formal burials of Māori dogs and puppies, further highlighting their significance. And these distributions represent significant environmental changes that Māori faced following the extinction of all species of moa, the loss of other bird resources, and the collapse of seal rookeries, as with the onset of the Little Ice Age and changes in society with increase in pa or fortification and horticulture as well. So I wanted to find out how kuri faced through the environmental transformative changes that Māori experienced, looking at North Island kuri assemblages with permission from Māori iwi tribe groups, the Auckland Museum, and senior archaeologists that I'm grateful for who are caring for these collections. Specifically, I was interested in seeing how kuri, diet and stress regimes might be altered. And this leads into what teeth can tell us. So teeth preserve quite well in archaeological deposits. They often survive long after the supporting structures degrade. And I use non-destructive methods to identify dental markers. And these results can be integrated into multidisciplinary approaches, including isotopic studies to gain insights into dog husbandry and health, which we have recently contributed to, as you can see. So for my research, I looked at 8 North Island archaeological assemblages for various time periods throughout Māori history prior to European arrival. And one of these sites, Kohika, here, will be mentioned a bit more in detail later. And I analysed approximately 135 kuri individuals, and using the University of Auckland reference collection and age guidelines from pioneering work on kuri by Jan Arlo and Professor Jeffrey Clark, I was able to age most individuals. And the North Island assemblages consist of relatively young dogs. So I conducted the analysis on the premise that dog developmental stages like birth and the post-weaning period in dogs, along with environmental conditions such as seasonality and disease, shape the life history of individuals and are reflected in their teeth. Additionally, stresses that can impact an individual can be accelerated or inhibited by animal management practices and changes to regional or local environments. So I looked for evidence of cavities or caries and dental plaque or dental calculus, and evidence of periodontal disease, tooth wear, as well as wear can come from scavenging in gritty or sandy soils. And I considered a condition known as linear enamel hyperplasia, and this results where the formation of tooth enamel is interrupted. For example, when an individual has a poor diet, disease, or a bad infection. And this was the first time that this condition was applied on kuri teeth. So my results indicate that there were no caries or calculus in any of the specimens. This contrasts to analysis on archaeological Hawaiian dog teeth where caries is present and illustrates dietary and oral health differences in Polynesian dogs. There's no severe instances of periodontal disease in the North Island kuri, and that's consistent with previous research where this condition has only been found in a few individuals from the South Island and associated with extreme tooth wear and diet. And overall, the North Island dogs exhibited quite light wear, with some dogs from Kohika having more pronounced tooth wear, even in young dogs. And this suggests that variation in wear reflects differences in the environment rather than age. And so just to show you the differences in the skulls, on your left is an adult dog from Oruarangi, one of the sites, and that individual has very little wear. You can see the cusps are still retained, compared to one of the dogs from Kohika, just showing you that extreme wear, and that indicates regional environmental conditions. So a more direct indicator of physiological and nutritional stresses in both dogs and humans is enamel hyperplasia. In the sizable northern Aotearoa assemblages, it occurs in low frequencies, being found at one early site and 2 off the late sites. The 2 types of hyperplastic defects observed were pit-type defects that you can see on your right-hand side. Your left, yeah. Here, a pit-type defect, and linear enamel hyperplasia, bands of reduced enamel on the teeth were the types identified. And the results suggest overall healthy dogs from the analysed assemblages. So I also looked at the timing of crown formation for the development of tooth enamel and the subsequent timing of enamel hyperplasia. And the results indicate that incidences of enamel hyperplasia in North Island dogs were concentrated around the timing of post-weaning. And no defects observed on both the deciduous and adult dog teeth developed in utero or in the womb, suggesting good maternal health of kuri. And that means, in other words, that the mothers of these individuals analysed were healthy and any physiological stresses occurred after those individuals were born. So to gain a better understanding of the wider context of kuri in diet in the North Island, I looked at available work on dog coprolites. And there is dietary variability across the North Island sites that is possibly related to both anthropogenic and natural environmental conditions. Fish bone, scales, and fish teeth are present, you can see, in the kuri coprolites throughout all time periods, and that's consistent with historical accounts, which frequently mention that dogs were fed fish, and dogs will often self-forage for fish. Other coprolite components help explain the moderate tooth wear patterns observed in our assemblages. The extreme wear observed in the Huka dog teeth may relate to consumption of shellfish, along with ingestion of sandy sediments and possibly tough grasses, further highlighting this regional variation within the North Island. So coming back to insights on dog husbandry, my research considers these 5 dental markers that are tooth wear, periodontal disease, caries, calculus, and enamel hyperplasia. And these markers provide insights into dietary change and overall health in dogs and indirectly inform on human management practices. And the study shows that dogs remain an integral component of the ancient Māori world prior to European arrival. And also the quality of dog diet and health is generally high, but there is some geographic variability. Specifically, there is little temporal variation, but the Huka site stands out, pointing to differences in kuri diet, experiences, and husbandry practice. Also, there are some differences in the diet and oral health between kuri and Polynesian dogs in Hawaii, for example, and that highlights further variation. So studying the Māori dogs adds to emerging global conversations about humans and dogs being the oldest animal domesticate and their place in indigenous communities around the globe. The Aotearoa case offers insights into dog-human relationships at an intimate level with the absence of many other domesticated animals and provides another lens into the world that Māori occupied with their canine companions. So I'd really like to acknowledge tangata whenua, as particularly the local iwi who supported this research and the wider archaeological community, my supervisor Melinda Allen, who's here, and just maurururoa, thank you so much for listening. Ngā mihi nui kia koutou katoa.
Our next talk is by Cedric Poulston, who will be talking about the role of marine versus terrestrial diet inputs on 4 Polynesian islands.
Hello. My name is Cedric Poulston. I'm a demographer interested in human behavioral ecology and other topics. And this is a project that was done with several other co-authors and a cast of hundreds of thousands, actually, when you really dig into the data that we've borrowed and used here. Other people working on this project: Jennifer Kahn, who just introduced me, you've seen plenty of; Nick Belluzzo, who is a former graduate student of Jenny's; Oliver Chadwick, soil scientist; and Pat Kirch, who you guys all know and love very well. The framing of this concept of this project is one of a natural experiment, and natural experiments are done where you're interested in a question, but you don't necessarily have the ability to manipulate the environment. So we have in the Pacific the opportunity to do some wonderful natural experiments, observational sort of studies, and one of the things that is most variable around much of the rest of the world, but we have reasonably good controls for here, is that most of the remote parts of eastern Polynesia, of remote Polynesia, were colonized by a single group in a relatively short period of time. So that eliminates a lot of the variability you might see on a continental system or in a different system. So one of the things that defines these groups, these people, these populations, as they came out, is something that has been mentioned a few times, is this idea of transported landscapes or portmanteau biota, which is basically the material things that people brought along with them, the foods that were important to them, and this was consistent in I think in almost all the cases that at least the people have talked about. These are the things that are important to people. They were always taken with them, and they so the fish, which are a considerably important part of the diet, are there when you arrive, and that's a resource that you exploit, but you bring your own sort of key foods, and these are central to the definition of meals. They're central to the, the definition of meals and to self-identifying, which include the starchy crops that were so important to the Polynesians, pigs, and also several other animals and plants. So those are the things that are relatively constant, the starting points. What are the things that vary in this natural experiment? Well, the islands that these people landed on. And this, if you look at the sort of the breadth of Polynesia, you're looking at a tremendous area, number one, 70 degrees of latitude, roughly in its extremes, and temperate to tropical variation, one way and then the other way again. And the result, or at least that's part of the argument, what the variability that you see on these islands in some of the social features, the ways that people prepared food, the foods that were important to them, and the population densities that occur on these islands. The question that we're asking is, what's the relationship between the nature of these islands themselves and some of the social features that we see? So motivating question, as we're sort of narrowing down to one part of this, is can we identify and understand interactions between ecological factors and population size or density? Specifically, and this is what we're going to be looking at today, does the balance of terrestrial versus marine resource availability have an effect on the maximum population densities? And so here's where we're going. Now, we're going to look at 4 islands, all of which have been introduced so far in the last couple of days, and I will just briefly go over them. You guys are familiar with our next door neighbor, Moorea. Relatively large terrestrial space, young island, 134 kilometers squared, and relatively young in this sequence of islands that we're looking at, about 1.5 million years. Relatively small, fringing reef compared to some of the others that we're going to see here. Further down the chain, Maupiti, this island much more subsided, just a small volcanic cone remaining, a fairly large lagoon around, and a lot of motu area, which is coral based. It doesn't have the same agricultural potential as the volcanic center. So smaller, considerably smaller than Moorea as it's subsided into the ocean. 4 kilometers squared for that volcanic center, and about 12 kilometers squared if you add the motus. And age is about 4 million years old. And then the other 2 that we're going to look at is in the Gambier complex here, the Mangareva and Taravai. They're extensive, huge lagoon around, extremely rich, and differing in size, about 3 to 4 between Taravai and Mangareva. Mangareva about 17 kilometers squared, Taravai about 5 kilometers squared, and these are about 6 million years old. One of the first things that we did was we took classifications of different Polynesian agricultural zones, and we did our best to apply them to these to all of the area on these 4 islands. And these agricultural zones vary in their productivity in terms of the numbers of kilocalories you could get per area per unit of time. Wet taro being the most productive, one of the most prized, usually one of the first things that people went for when they arrived on it. This is a highly prized food. down to upland semi-cultivation, which are famine foods found in poor areas, could probably realistically be thrown out of this this analysis because they really just the labor required to gather these foods probably doesn't make it worth it in a lot of cases, and could probably just as well be eliminated. And maybe even below this, you've got a zero, a category where we deemed, this either was inaccessible or the soils were so poor that nothing productive could be grown. How did we identify these zones on these particular islands? We used primarily two methods. One is the substrate, the soil. This was done from soil maps, and we looked at slope, which is done from digital elevation models in a GIS context here. The productivity estimates for these various zones were taken from a number of sources, but we relied heavily on Pat Kirch's 1994 Wet and the Dry, and Hamilton and Kahn 2007, which study of Punhu Bay on Moorea, as probably our most important sources. I'm just going to show you an example of what one of these, the zonation for the island of Moorea, and what we're looking at here is colors representing the productivity that you could get in a kilocalories per hectare per day, and the yellow zone in these sort of stream valleys is where you're seeing the richest. So we did this for each of the 4 islands, and then we multiplied the productivity for each zone by the area allocated to each zone, and we come up with which in the first column of this table here. So you've got the islands on the rows. First column is the total productivity. If you add it up in a single day, what you hypothetically, under these assumptions, how much food you could sort of pile up to allocate to your population. That's that first column. And you can see right away that Moorea is in a different category than the other islands, and that's largely a function of its size. But so 168 million kilocalories per hectare, or total across the island per day. Maupiti is about 6, Mangareva about 11, Taravai about 4. Next 2 columns, measures of area and agricultural area, refers to everything but semi-cultivation or an unproductive land. So we're just saying how much of this space, the area of this land is dedicated to effective production. So then you end up on the next column over is a population estimate, and the way that we came up with these population estimates is a sort of back of the envelope computation. It was used by Bayless Smith, 1978, and others, where you assume something like, in this case, 2,700 kilocalories are the requirement per person per day, and we're assuming 30% of the total production is lost and not available to the population. That may be in ritual use, that may be in wastage, that may be in crop failures or whatever. But it's a formula that's been used before, and you can criticize it, but it has been used. It allows you to compare across. And the numbers that you get here in these last few columns, the next 2 columns are measures of density. One, this total population as a function of the total land area that's available, and the second, the density as a function of just the productive agricultural land, which is a good way to sort of compare, especially when you've got large areas that are not available for intensive agriculture. So, and I think most people would argue that these numbers are probably on the high end. They're not too far out of the range, but and we'll come back and revisit it if we get the chance here. But these are the numbers that we come up with. So this is a representation of what we would consider the classic carrying capacity, minus some correction factor for loss, used over and over again to ballpark what we have. What we have here is probably a much more detailed analysis of what that agricultural potential is, and that may increase the numbers of total productivity compared to what you may have seen in the past. So the other thing that I would say is that the kilocalories, the food that we're talking about in this total productivity, is largely the starchy agricultural products. Good Lord. That make up the bulk of the Polynesian diet, the typical Polynesian diet. 85 to 90% of those kilocalories are coming from carbohydrates. So does that matter? I mean, do we need to worry about the other components of the diet, the macronutrients, the 3 macronutrients that are important here, or that exist: carbohydrates, proteins, and fats. Carbohydrates, we're pretty sure we got covered. Fats, we're not too worried about. Carbs and fats can be exchanged for each other to some extent. But protein, you have to get from a limited set of sources. Where's that protein going to come from? Do we need to worry about it? Is the question that we're looking at here. So in the Polynesian context, you get protein from terrestrial sources that pigs, birds, dogs, and rats, eggs, plant proteins, and then there's a host of marine resources that may be available to you to certain degrees, depending on where you are. And these include sort of inshore fish, deep water pelagic fish, and a number of other sources, including mollusks, and just in occasional cases, marine mammals. So we have these, the excavations that led to this table were mentioned yesterday. Actually, I think all of the sites were discussed at one point or another. There are sites on Marae and Mapiti that were excavated by Jenny Kahn and colleagues, and categorized by some of the people in this room in their dissertations and all the rest of that. And then Pat Kirch and company did excavations on Mangareva and Taravai and collected faunal remains. Those faunal remains were characterized and later on converted into estimates of biomass, which is great and it's interesting. It's really cool. But what we're interested in is we want to be able to compare these relative numbers to the absolute numbers of the agricultural. So we're looking for sort of ratios here. So how do we get from one to the other? And just one of the things that I would point out before we move any further is the marine signature on Mangareva and Taravai. And those data were summed because we didn't have that much variability. We didn't have that many layers, I would say, to characterize in the Mangareva and Taravai cases. So we're limiting ourselves to the 1400, 1800 period, and we're excluding elite sites, which can be skewed in terms of their faunal assemblages. So how do we turn the relative biomass into an absolute number? Well, we turned to the literature on subsistence fishing and found a number of sources. One of the best was probably this publication, Newton et al., 2007, in which the authors looked at approximately 79 studies of subsistence fisheries in similar contexts to the one we're talking about here, and found a median fish yield, live weight of 3 metric tons per kilometer squared of productive reef per year. So, and because we're looking at, and these studies, so that's the ballpark of what we're looking at, and they range. There's a very, very wide range across there. 2 minutes, all right. We ended up choosing to go with 5 metric tons because that was believed by at least most of the people involved here to be the sustainable figure. So we want to compare as close as we can, apples to apples here. So what we did is we characterized from aerial photographs the reef, productive reef, to estimate the productive reef, and that includes areas that are submerged, but not submerged deeper than 30 meters. So deep water not included in this analysis. So Morea we included, did like Apunu and Cook's Bay, not included. So we've got the graph on the bottom, or the table on the bottom, that shows what those values were. So then we can add this stuff up. Now we have an absolute measure of the reef fish that we can use to scale all of those other faunal remains. And again, I mean, it's a leap of logic, but I think it's a reasonable one, and it's the best we can do at the time, at the moment anyway. And so what's our result? You get this really, really strong skew when you add up all of these caloric resources. And so if you look at Morea in particular, 99.6 of its total caloric availability under these assumptions is coming from terrestrial inputs. And the animals, the terrestrial animals are showing up in the terrestrial total here. The marine inputs are only 0.4%. So like what? Like how are we gonna deal with this? So Mangareva and Taravai, you have a very, very different picture where 15% marine, 13.1% in Taravai. So really different picture here. So why do we get this skew? One of the interesting things that came out is the difference in productivity per unit of area in terrestrial versus marine environments 150 times. So a kilometer squared of agricultural land is 150 times as productive as a kilometer squared of productive reef. And that's actually what's driving a lot of this stuff. So the first table here, I'm just about at the end, is the table that we looked at before. That's the upper table here. And now the second table, the one in the middle of the screen there, the second one down, is an updated table. We've included the additional sources of caloric input, and also in the circle there, we ask the question, what happens if you assume, if you consider protein limitation? So in that skew, you got 99.6% of your calories are coming almost exclusively from agriculture, and you're only getting, I think, what is it, 4.5% of those calories are dedicated to or coming from protein. You add up all the protein sources. How many people could you support on those protein sources that we've got a full tally of? And Mo'orea's numbers drop from about 43,000 to about 23,000. And that was only true on Mo'orea. The other 3 islands seem to have enough protein to not run into protein limitation. So we don't have time to go into it, but I'll leave you with this question. Under what conditions might a population fall short of its theoretical maximum? So you're still looking at the densities. And I would argue, and I'm sure other people would argue that those are probably pretty high, and maybe even, well, they look like they're high to me. So why might we project theoretical densities that may never have been realized? And there are a number of ways to answer that question, but we don't have time to go into it. So, and so just coming back to the last 2 things, coming back to our motivating question, the maximum potential population, a measure of carrying capacity, is largely driven by terrestrial agriculture, at least on these 4 islands, at least given our assumptions going in. And protein limitation cannot be ignored in cases where you've got a very high agricultural potential and probably accompanied by not extensive lagoon areas. So, and I would leave you with that. Thank you guys so much. I appreciate it. And the organizers, through the COVID years, that we're here. Thank you so much. And the translators, bravo, thank you guys. So.
Our next paper presentation is by Anna Gosling, and it is entitled How the Chicken Crossed the Pacific.
Okay, so I feel like a little bit of a fraud here because I'm a molecular anthropologist, and most of the work I do actually involves understanding human health in the Pacific, and particularly metabolic disease. But it turns out that to understand this, we really need to understand more about how people distributed across the Pacific, the relationships they had with other people, and sometimes that involves looking at the animals and other critters that they carry along with them. And this actually also suits me quite a lot because I really love chickens. And so this has allowed me to combine the work I love doing at work with something I love personally. So we'll take you on a journey looking at chickens in the Pacific. So when you look at archaeological sites throughout the Pacific, chickens are actually very broadly distributed. They seem to have arrived very early on, and you see them in some of the quite early Lapita sites up here in Musau, and up in Wattam. And they also distributed down with the Lapita people down to Vanuatu very early on. We see them down in Tonga in this time too. So they were part of the early kind of transportable landscape that we kind of talk about quite often when we're talking about Lapita, but we also see them a lot in later sites as well. They disperse quite rapidly out throughout all of remote Oceania, well, mostly, apart from down here to New Zealand. So you do have this distribution of domestic animals across Polynesia, where you do have some of these domesticates that we think were transported missing, like pigs, which we don't see in certain islands. We do usually see chickens, and we see chickens very abundantly. So it seems like they're filling an important niche where protein is needed. So, yes. So this falls into the commensal model for tracking human mobility in the Pacific, which Catherine talked to you a little bit about yesterday, where we can use the genetics of these animals that were carried along throughout the Pacific by people. Obviously, chickens can't row a canoe and can't get to places by themselves. So we can look at the DNA of these animals to look at interactions between people in the past. We'll take you through a quick journey, a personal journey of chicken variation in the Pacific. You can see, obviously, we've got a lot of chickens in Vanuatu. They're quite phenotypically different these days. Lots of different chickens, very pretty, A+. We also see them, I've also obviously been to Rapa Nui. Again, we see chickens throughout Rapa Nui. And here, Rapa Nui people are obviously very close to my heart because chickens here were clearly quite important prehistorically. And you see the construction of these hare moa, or houses for chickens, in the past. And we can see some contemporary chickens here using these hare moa. And we've also got some quite cool dendroglyphs as well, where you can see here they have carved a few chickens on this piece of rock, and I think this is a hare moa at the top here. So the fact that people have gone to the effort of carving this into a rock clearly suggests that chickens are quite important. And again, we've got local chickens here as well. Here are some I met the other day. Very cool, love them. Anyway, why should we study chickens? I've kind of covered this a little bit already. They're part of this transported landscape taken around the Pacific, not only by the Lapita, but people moving out into Polynesia more recently. Chickens can't move themselves between islands, so they rely on people to do this. So it can tell us more about people's movements and their social connections in the past. It can also contribute to understanding of domestication processes more globally. So the process of getting wild animals and turning them into things that are a bit more useful to us, so that we can grow and travel more easily. The problem is that modern chickens are, well, they do a lot of breeding with each other. So we've got a lot of phenotypic variation in contemporary Pacific chickens, but we don't necessarily know whether this is something that if we went back a few hundred years, this would still be the case. And this is because we've got the early introduced chickens, but we've also got chickens that were introduced by Europeans, and these guys do not interbreed. I mean, chickens, they make babies all the time. Anyway, so this makes it quite difficult because we can't really look at modern chickens to untangle what was going on in the past, because we've got all this admixture, just like we see with humans, with chickens introduced in the last couple of hundred years. So this means that for taking a commensal approach, we really need these ancient chickens and the bones of these ancient chickens to understand paths of migration and into island contacts and things. So this has, to a certain extent, been done. There have been a number of ancient DNA studies which have generated partial mitogenomes, so a couple of hundred base pairs of ancient DNA, to look at this specific question. And what Story et al. Found, that working with Lisa about a decade ago or so, was that it looked like there were a couple of distinct chicken lineages, which they were calling lineage D and lineage E, that were distributed out across the Pacific, and these were linked to different temporal introductions. And so it seemed that we had a relatively early introduction of haplotype E, and then a secondary later introduction of haplotype D, which comes in a little bit later. And this is kind of cool because it tells us a lot about connections with people. It also, around the same time, some chicken bone was found in the site of El Arenal off the coast of Chile. These came back with it in a nice secure archaeological context with radiocarbon dates that dated pre-Columbian, so it definitely looked like these guys were introduced by probably Pacific voyagers coming along the coast of Chile, probably picking up their kumara and things along the way, and being left behind. The DNA sequences of these chickens matched the ones that were being found in East Polynesia, so belong to that what we were calling haplogroup E. And so it seemed like a really nice story of Pacific chickens being introduced to South America. However, these results did become contested back in 2014 by a group from ACAD, who suggested that all of the haplogroup E chickens were actually a result of contamination, and that all of the Pacific chickens in the ancient times belonged to haplogroup D. And so this is actually cast a really long shadow over Pacific genetic studies, and it's had quite a lot of repercussions in other studies that have happened since. But quite interestingly, a Chinese group, Miao et al., they've actually found in their study of small hypervariable regions of DNA that you can't actually tell the differences between haplogroup D and E1 based on that particular length of DNA, based on the mutations that you see in there. So while we now know that they were right, and that Pacific chickens do all belong to haplogroup D, it doesn't necessarily exclude the possibility of two subtypes of haplogroup D within the Pacific. It also does suggest that we should be all transitioning to technology that is capable of detecting contamination, and so we'll cover that in a second. So what we have done is move on to sequencing a larger portion of the DNA. So you can see here in this figure that we've got the control region. This is what the old studies were looking at. It's only a few hundred base pairs. But for our study, we have now sequenced the whole mitochondrial DNA. So that's the maternally inherited 16,500 individual letters inherited through the mother's line. So this gives us a lot more resolution. It allows us to figure out a lot more specific lineages. And also the technology for doing this is a bit different. So we were not, obviously, convinced by this contamination argument for the previous results. So we have used the latest technology to check this out. This is the next generation sequencing. Next generation sequencing has a lot of advantages over the old Sanger sequencing methods, which was the method, the technology that was used in both of the previous studies I was talking about. And one of these is that it sequences all of the DNA molecules that are present within a sequencing library. So this includes the DNA from the chicken, it includes DNA from the environment, and it also would involve any sequences that are coming from other contaminating sources, such as reagents. From the experimenters like me, we can see them, they're all there. And so this means that if there was DNA from chickens contaminating our ancient chickens, we will be able to see that within the data that we get back. So we have prepared DNA from 54 chicken bones throughout the Pacific, including New Caledonia, Tonga, Samoa, Hawaii, where else? The Marquesas, as well as a historic site of the Isla Mucha off the coast of South America. We have successfully sequenced only 12 ancient Pacific mitogenomes and 2 historic South American sequences from these chickens. And I will note that generally ancient chicken DNA from the Pacific is very poor, which is why we've got such a poor success rate. Compared to some of the other animals, including Kiore, like Catherine talked about yesterday, the recovery of DNA from chickens is pretty sad. Anyway, these are the results we've got. I've just made it into a network which shows the relationships between different samples. You can see that some of these, so the coloured dots are different individuals that have been sequenced. Some of these, like these ones up here, are ones that have previously been sequenced by other labs and which are available in online databases like GenBank, whereas our new results are down in this area down here. So you can see that all of our our Pacific chickens, which are these green ones and these dark greeny ones here and the pink ones, they all belong to haplogroup D1. And they cluster broadly into 2 groups. So we've got what I'm just calling group 1, which is over here, which includes some ancient chickens from Rapa Nui, some ancient chickens from Hawaii, some modern Marquesas and Fijian chickens, and a few modern New Caledonian Vanuatu chickens. They're clustering this group 1. And we've got this other second group over here, which I'm going to call group 2, which is separated by about 10 individual mutations, making it a quite distinct group. And this includes a few, well, we've got only one Tongan chicken here, which dates to the Plainware period, but we have a few slightly less good quality, but definitely very similar to this specific chicken. Samples coming from 3 different Lepida sites in Tonga. We've also got these 2 chickens here, which are from Micronesia, so from Yap and Chook. They're also quite similar, and they're in this this group 2 group. And we've also got a few Hawaiians here, so I think it's 3 individuals. So it does suggest that we do have two chicken lineages, which reinforces the work that was done by Alice and others back in the earlier 2000s. And they are differentiated by a number of variants, so that these are chickens that were brought along to where they are, pretty much as they are. It looks like we've got some sort of temporal control here, where we've got these early Lepida and Tongan plainware chickens sitting in this group 2 group, compared to these predominantly East Polynesian chickens over here. So that's quite interesting. And to just reassure you, we can definitely tell the difference between D and E chickens using these whole mitogenomes, because we've got these historic samples from Isla Mucha, so in Chile, sitting up here in a completely different part of the network, showing, you know, we can definitely tell the difference between E chickens and D chickens, so it's not contamination. So, just re-sum that up. We do have all ancient Pacific chickens belonging to haplogroup D, and if we go back here, we've got a probable origins. Haplogroup D arises up in the Philippines and the island Southeast Asia area. So the Pacific chickens that we're seeing in archaeological sites probably arise in island Southeast Asia, which is what we know about chicken domestication anyway. We have got 2 distinct lineages separated by about 10 mutations, and we can definitely tell the difference between E1 lineage chickens and D lineage chickens. And we can confirm, having looked very closely at all of our samples, as well as negative controls that were prepared alongside the samples, that we have no contamination within these samples. So we're pretty certain that what we're seeing is right. We've also re-investigated the South American connections question. Sorry. Yeah. We've unfortunately not been able to get the original bones for that LRNL result that we talked about earlier. They've not been allowed to come back to us for retesting, but we've been sent some recently excavated bird bone from new excavations at LRNL to try and confirm whether or not the original findings were legitimate. Unfortunately, the bones that were sent to us were not morphologically identifiable as chicken. There were no diagnostic portions left on them. We ran them anyway, and unfortunately, they're not chicken. The specimens belong to a great tinamou, which is a small galliform endemic to South America, but they had very, very good DNA, and they had very good depth of coverage, and they were also clean of chicken contamination. So we're pretty confident that if we were able to get the bones back, we would hopefully be able to get a result. And tinamou and chickens, this is just an alignment of the sequences, are very, very different. So you can see at the top here, I've got the chicken reference sequence. We've got the 3 original chicken sequences from LRNL that were online. We've got the reference sequence of a Chilean tinamou, and we've got the 3 bones that we sequenced last year. And you can see that they are very, very different. You cannot confuse a chicken with a tinamou. So fingers crossed one day we can get those chicken bones back. We've also, so what's next? We've sequenced some genome-wide data from 4 of our ancient Pacific chickens, including the Lepida Tongan plainware chicken I talked about earlier, a couple of ancient Hawaiians, and one of the ancient Yapese chickens. These are still undergoing analysis, so we'll hopefully be able to tell more stories about what is going on soon. And just a note, as I've said multiple times, I really love chickens. They're my absolute favourite. They're fluffy, cute, lay tasty eggs, like keeping them as pets, like studying them. So if anyone has some nice archaeological chicken bones lying around that they want analysed, I would be very happy to receive and analyse your chickens for you. Anyway, that's my talk. Mahururu. Thank you.
It is time for lunch. Yep. Oh, sorry. Yes. We have 20 minutes of discussion before lunch. Yes, I was jumping the gun there. Okay, we have until 12 noon, so why don't all the presenters come up and we can see who in the audience has questions for them.
Thank you. It's just a quick question for Anna. What's the coverage like of chickens in islands Southeast Asia that allow you to say from the Philippines? I mean, how confident is that?
There's been quite an extensive sampling of contemporary contemporary island Southeast Asian chickens from quite a few of the different islands within the Philippines and Indonesia. It's somewhere in that area, but no ancient DNA as of yet.
Thanks.
I have a question, about chickens and eggs, but mainly for you, Cedric. So I have on your list of protein sources, you list eggs, and I've always wondered about that because on Rapa Nui there are so many very detailed accounts of what people brought for food. It's like 3 fish and 3 chickens and 17 sweet potato, and eggs are never mentioned outside of seabird eggs during the Birdman ceremony. And then I know there's a lot of loss of oral traditions on Rapa Nui, but that's a question. Like eggs, chicken eggs as a food source. Is there any evidence for that?
I'm probably not the best person to answer that particular question. I don't know. Pat, you look like you were, the reason I included them as a potential protein source, I did not have any particular information on whether on these 4 islands they were used. I would defer to Pat in this particular question.
Thank you.
Yeah, just a quick response on that. In my experience in several islands with fairly traditional subsistence systems like Tikopia, Anuta, Futuna, I never saw anybody eat a chicken egg. They do go for seabird eggs, absolutely. But I have no experience of seeing people eat chicken eggs. I used to get the kids on Tikopia to go in the bush and collect them for me so I could make omelets.
So maybe then, sorry, just a question for Anna then. Would it be a possible reason that I don't know a lot about chicken biology, obviously, but that the chicken eggs were just let hatch because the meat, the protein was worth more than an egg, an omelet?
I would say that they're not really managing the chickens probably that well once you get to islands, and so you just don't know where their nests are and how old the eggs are, and no one wants a rotten egg. And so I think they are mostly being used as a protein source, as meat, rather than as eggs.
Thank you.
One more comment on chickens. Polynesians had cockfighting also, so that's another reason for chickens. And the use of the feathers, right?
I have a question for Patricia about the dog work. Just super cool, super outstanding. I was really shocked by the fact that there's no calculus and no cavities, and that's just amazing, right? To see these dogs living directly, with humans and not having that. Some of our work, which I'll talk about tomorrow, looks at how there may be unique human microbiomes, for example, in New Zealand, and so. From a microbiological perspective, we know that calculus forms via very specific microbes. And so I guess I'm wondering about any sort of future work that you might be thinking about in the context of thinking about what microbes these dogs have and how that might be influencing their ability to make calculus in the context of a given diet, or whether or not that's something you've even thought about. It's probably way too specific.
Thank you. That's a really interesting perspective and question. I think Professor Judith Littleton, who was my co-supervisor, might have some takes on that. So I will discuss that with her. I mean, we did look at the Kohika coprolites, Melinda and I, and there are parasites in those coprolites, which are very interesting. And Judith had suggested there could be a link with why we saw some enamel hyperplasia there as well. But it would be interesting. I think the coprolites and thinking about the biogeomes would be really interesting. Thank you.
So I was wondering if you had Dog burial.
I was wondering if the dog burials samples looked different from the non-dog burial samples in terms of cavities and diet or anything.
Particularly the burials came from one area in particular that we've done some work with my other colleague Robin Kramer in the North Island and Matt Campbell. Surprisingly, no, they were quite similar, relatively healthy. I think one of the burials did have an incidence of enamel hyperplasia, but it was like one count. It was quite low, and that contrasts, I think, to what we were seeing in the humans with the human remains of Ko'iwi with they had calculus, I believe, and calculus and caries, and they had enamel hyperplasia. Sorry, but the dogs are not. So there's some differences there that are quite interesting.
This is a question for Iona. You said was it in, and I just want to clarify that in System, that's where you had the complete turtle.
No, that was
No.
well, there would have been complete turtles in there. We had all of the elements and a few individuals.
And the complete rats?
I believe so, yes.
Were there rat bones in the other parts of the site?
No, I actually did not
No.
find any. There were some where the non-enclosed cyst deposits, you could see a little bit of, rat gnawing, but there were no rat bones in
rat
any other
bones.
feature. Yeah.
Do you think it's
Interesting.
possible? I'll put it out there. that
Yeah,
maybe the
no,
rat
go.
body, the complete rat skeleton was placed in the cyst as well with a complete turtle.
I don't know. It's interesting to think about. I think System, it's hard to say because things were a bit like jumbled up. That's where we found the dog. There were fish and shellfish in there as well. So there's a lot of stuff happening with a lot of species. The rats were comparatively really well preserved, though. They didn't look like they'd been there for the entire duration of the deposit. And there were a few individuals as well. We had like juveniles and adults. So to me, it seemed like failed burrowing, but yeah, I guess it's impossible to say for certain.
Be good to date them.
Yeah, totally.
I have them in the lab for you, Lisa. They're ready for you.
I had a question for Jillian. I wondered if you could say anything about freshwater resources and where they're sitting in that diagram and how interesting they might be or possible to look at them.
I would love to say something about freshwater resources, but isotopically that is so challenging. So it's a very, very local signature, and we just have not done the amount of kind of ground truthing and sampling of the local environment to be able to narrow in on that. Definitely something that should be explored further, but haven't been able to get that refined a data set.
Thank you. That's sort of a heads up to all of us to pay a bit more attention when we're getting reference materials.
Absolutely, yeah.
But Jillian, didn't you think my 360 samples might have a little bit of a freshwater?
It's possible, Jenny. It's been a while since I've looked at those samples, but it's possible. I wouldn't rule it out, but we need to stare at that a little more closely before I will be like, yeah, freshwater.
Well, since I have the microphone, I'll ask a question or suggest maybe an idea. Have you, you chicken people, have you ever considered looking in other places, in other regions of South America for assemblages with chicken? I'm thinking especially of the island called Chiloé in Chile. They have lots of earth ovens and lots of oral traditions that sound Polynesian to me, and I've never looked into that, but I was thinking maybe if there are any excavations there, I might have points.
Yeah, we have. We did a survey kind of all the way down the coast in Chile to look for rat bone, chicken bone, and ultimately dog bone as well, because in the south around Chiloé. One, they have small potatoes that are the size of your finger that taste very, very sweet that nobody liked, which was very interesting for us. They have stone plank canoes that appear in the archaeological, that have been dated from, they pulled them from near complete from rivers dated to about 1300 AD. They wear kuri dog skin cloaks down there as well. So we've just done some dog bone from a site just off of Chiloé, another one of the small Chono islands. But there's no other chicken bone other than at El Arenal. And those chicken bones from El Arenal were morphologically identified as chicken as well. There's supposed to be there were 50 bones recovered from the site, but the archaeologist there says he can't find them anymore. We wanted to get more. And the ones that we processed them completely for radiocarbon date and two independent genetic ancient DNA analysis. So the actual bones are destroyed, but there were additional chicken bones recovered, but we haven't been able to get them. But yes, we've been looking. We did do some early historic chickens from Peru we had both the European chickens and what we called the early what we were calling E chickens from those historic sites as well. So I don't think that there were many of them. I don't think, but El Arenal is the only pre-Columbian site so far that has chicken bone identified.
Just follow up also and say that modern ancient DNA methods consume a lot less chicken bones, so we wouldn't run into the same problems of consuming whole chicken bones still nowadays these days.
Cedric, I had a question for you about, since your argument is so, especially for an island like Moorea, that the terrestrial system is so important. So if we think about stochastic variation, and I can give you an example, like say Captain Cook is there and he wants all the pigs, right? And so then you really have some pressure on the terrestrial system that you hadn't had before. How would that look in terms of your model?
Well, I think one of the conclusions you might come from is that the protein sources were were under pressure, and so you're in a cluster of islands, or at least you've got Tahiti just across the water, and if the possibility exists to trade back and forth, if you have an excess of carbohydrates, which are of high value, and can trade with your neighbors, maybe it's possible that you're releasing some of that pressure, and you end up, being able to realize more of the carbohydrate potential that exists on your island by getting protein resources from somewhere else. But to answer your question, if there was something, a low probability event that swept away a huge fraction of the protein that was available, either by force or in exchange for something that was considered more valuable, you can just imagine the pressure that would put on the diet of everybody living on that island. And I'm not sure if there are sources, if we have any information about what happens following, some of those visits. But Pat, sounds like he may.
Well, as you're talking about that, I'm just thinking of the cases, several of them, where protein sources were eliminated, particularly pigs and dogs, right, where we have them earlier on in the sequence, and then they're presumably purposefully eliminated. And I've argued that that was because of trophic competition, but in terms of what you're saying today, then that suggests there might be more stress in terms of dietary stress. Once you
I
remove
would say it would
those.
depend on the context, really, because Moorea was the only place where the protein limitation was a serious consideration. So in most cases, protein might be either considered a luxury or.
But I'm thinking of Mangaya, where the reef is extremely small and limited, so your marine resource would be even worse than Moorea, and they eliminated both pigs and dogs. But interestingly, 8 tons of rats, right?
and maybe that's an illustration of the same, you're trying to solve that problem, and you found a different solution that people on other islands did, and complete speculation. But it might be worth doing that same kind of analysis, doing that there, looking at the diet breadth, at least potentially.
We have time for one more question from the audience.
I was just going to ask, how does pelagic factor in? And is there representation of pelagic fish remains in archaeological deposits at a higher frequency on the RURUA, for example, when you have access?
It's been a while since I looked at those in detail. I mean, there were pelagic fish. Sometimes some of them could be caught near shore at either some phase of the life cycle or not. But they definitely existed. I can't remember what the distribution was across the 4 islands. But Jenny may be able to speak to this.
The 350 site that I talked about yesterday, the pelagic is highest in the colonization phase, but then it drops out like most other places. So it does not continue to be exploited. So there wasn't that sort of like prey switching as an answer to that specific marine context. I think everybody's probably ready for lunch, so why don't we give everybody a hand?
Just a quick note here. So we're inviting the participants to go have lunch at the same place as yesterday. It seems like it's starting to rain a little bit, so we're going to organize some rotations with a few cars. So if some of you want to wait upstairs, we'll come and pick you up and do a few trips.